What'd you say? Assay is a word that means a type of experiment where we're like measuring something specific, and I'll talk more about it later. But so like an activity assay, for like an protein that normally like cuts DNA, you could do an activity assay to test how well it cuts DNA. And so assay is just one of these many words in biochemistry that can that we tend to use a lot without really thinking about it. But if you don't know what it means, then you can feel really lost. And I have this thing in my head, like one of those negativity bias things, where I have this really strong memory for words that I didn't know what meant when I was younger and was really confused but was embarrassed to ask about. And so this whole like negativity like thing is really bad for self-confidence, but it's really good when it comes to teaching people because I remember what it was like to be in that situation, and I have this like list in my head of remembering the words that I didn't understand, and so trying to make an effort to really explaining these terms well when I use them. And so with a lot of like science communication efforts, they tell you to like avoid jargon, so like avoid these like technical terms, and just use the more common language. And so this makes sense in the terms of when we use this common language, it's not that we're dumbing things down, we're just kind of translating. So it's not that people don't have the skills to understand these terms, it's just they haven't heard these terms before, so they don't know what they mean. But instead of just using the common terms, I like to actually give you the jargon and the common word explanation. So this way, if you see the jargon in the scientific context or whatever, you won't feel like left out and you'll be able to follow along, hopefully. So that's one of my main goals with like the bubbling biochemist, which is why I started this on my blog. I have a glossary section, and so if you're actually like reading the articles on my blog, you'll see underlines when the words are in the glossary, and you can mouse over it and it'll show you the definition, and then you can click to get to a further definition. And so I thought I would take you through a few of the words that I used to be the most confused about, and their entries in my glossary and what they mean and that sort of thing. And so I hope that helps, and let's take a look. Let's start with the word assay. Other than the source of some embarrassing Siri-assisted voice-to-text texts, what does this word mean? Basically, it's just a fancy word for an experiment in which we're measuring something, like some property or quantity of a thing. And so that's kind of vague, and so really this term means a lot of different things. It's basically some test where you have some sort of readout that tells you about how much of something there is. So it could be like the amount of a protein or the amount of a drug or the amount of a certain chemical. So you often hear it used in the chemistry and that sort of field when you're trying to measure the amount of a substance. In biochemistry, we're often using it to talk more in terms of a functional property. So like how much activity does an enzyme have? And so an enzyme is typically a protein that helps speed up a reaction. And so you might be doing an assay to measure how much reaction speeding up power that enzyme has. So basically for an assay, you're typically like changing one thing, or you're introducing some substance that that the thing that you're measuring will be able. So if you have like a protein and you want to measure its ability to cut DNA, you might add some DNA and then measure how well it cuts. So you need some sort of readout as to how much of the DNA is cut. If you have you're doing like a binding assay, you might have a protein and like an RNA molecule that you're seeing if it binds to, and so then you need some sort of readout as to whether it binds. And so this can be measured by something like a slot blot or an EMSA or some sort of fluorescence readout. So with assays, you can have things like endpoint assays where you're just adding something, waiting, taking a measurement at the end. Or you can have kinetic assays where you're measuring over time. You often hear about like high throughput assays. So that's basically usually using like robotics where you have these plates with tons of little wells and you're doing a lot of experiments, like testing a lot of different samples at once. You can also have multiplex assays where you're testing for multiple things in each sample. Assays can be qualitative, so basically just like there's some sort of yes no readout, or you're something like that where you just kind of getting a sense, but you're not getting like legit specific numbers. Whereas with like a quantitative assay, you are doing those specific getting specific numbers. Then you can have things like a semi-quantitative assay, which gives you numbers, but it's not so reliable, I guess, kind of in terms of those numbers. So sometimes you can think of like a Western blot as being semi-quantitative because you can kind of base it on the amounts of like a protein that you like normalize them to a loading control. So you have one thing in the sample that should be the same no matter what the cell type or anything. And so then you can use that. So compare what your band looks like to that band in the different samples, but it's not like a true one-to-one comparison. So we call it semi-quantitative. So basically, yeah, assay is not very easy to describe because it can mean so many different things. But when you hear an assay, basically just think of some sort of experiment where you're trying to measure something. Another one that can be really confusing is the word residue. So we use this word a lot, especially in structural biology, where we're commonly dealing with what proteins look like. And so proteins are built up of protein letters called amino acids, and these link up through these peptide bonds. And so amino acids have this generic backbone part, and then this, which has an amino group at one end and a carboxyl group at the other end, and then in the center is the central carbon. And sticking off of the central carbon is a unique part called a side chain. And this is really where the cool stuff comes in. So this is where you have, there's 20 unique different common side chains, and these have different properties, size, charge, etc. And so that's going to influence how the protein can interact with other things and with other parts of the protein, so how it's going to fold up. When you link amino acids, that unique side chain is going to stick out kind of like a charm from a charm bracelet. But that amino group and the carboxyl group combine when you link up the amino acids. And when they combine, what you're left with is what we call the residues. So they're no longer amino acids because they don't have that amino group and that acid group, because those groups have combined in this peptide bond. So instead of having amino acids, you have this polypeptide. And so now each of what used to be amino acids are now residues. So typically, so when you're referring to like the protein letters on their own, so if it was just like free floating, it'd be an amino acid. But once you link them together, then each individual, what was an amino acid is now a residue. And so often we just call them amino acids for simplicity or just habit, I guess. But technically, once they're in the context of a protein, we call them a residue because it's the residual, what's left over after you combined them. And the last word for today I want to talk about is aliquot. And so basically, aliquot just means like a little snack size or single-use portion of something. So if you make purify a bunch of a protein, and you don't want to have to keep like freeze thawing it, which isn't good for proteins, you can make little small samples so take little amounts of the protein before you freeze it and stick them in little each in separate tubes. And this way, whenever you want one to use the protein, you just have to take a single tube out and thaw it and use it right away. And you don't have to worry about keep freeze thawing, which your protein won't like. So I hope that helps.